Okay, so you all turned in your assignment and everything. Now, I assume you're all working on your final paper topic and description, right? Making progress on that. So what we really need is there is basically a topic, right? You want to, what's the idea that you're trying to study? But also you want to send like around like I put like in 300-word description of what the idea is, why you want to study it, and if you found a couple of papers already, you want to mention those and explain what the motivation is. Okay, any questions about that? Are you all clear on it, like working on it and yeah, like no questions? I mean, like, as I said, you could always email your TAs and ask them questions and bounce an idea off of them and see if they think it's a reasonable thing for you to do or not. It's manageable or not, all those things.
Are there sample papers on Canvas or no?
For the final paper?
Yeah.
Yeah, there is one I put there in the syllabus and everything on there must be a file here. So it's on the syllabus or modules. Where we describe what the paper should look like and stuff. And then I put what's that? Yeah. So let me go to the syllabus so that way it's easier to see it. So I didn't save a copy, but it's available online. So for the final paper, yeah, this is the example for you to see what the format looks like and what the general organization is. Obviously, this is not your topic, so it would depend on the topic and everything. And also, at this point, you don't want to worry about the final paper format and stuff. You just want to worry about having a topic and stuff like that. Yeah. For now, worry about that. But this is a nice sample for just what are the different components that you need, basically title and your name and the department information, abstract, a short intro probably before you go into your organization of what different relevant themes are in your topic. Okay. Other questions about the topic? So that means that you're all thinking of a topic or you already found something and you're working on it, right? Okay. I think it's due by Monday. I think next Monday around midnight. So try to send that in. Today, we're going to talk about behavior in general, talk about the actions. We'll talk about different brain areas that are involved in it. I'll talk a little bit about technology too, about brain-computer interfaces that are inspired by this research and try to help people. So first we start with eye movement behavior. And before that, I want to make a distinction between covert and overt attention. This is something that I alluded to in the last lecture, but I didn't really get into it. If we move actually something, a muscle, right? It could be your eye muscles or your entire body or your head in order to attend to something, right? Then that becomes overt attention, like it's in the name, right? You're not doing it covertly. That's what it means. If you're not doing any of those things, then it's a covert attention, right? So covert attention refers to our ability to attend without moving our eyes or turning our head or doing anything else, right? It could be that you are in a party and you're trying to listen to someone, but you're not moving, you're not turning your head to hear them better or anything, but you're just trying to covertly attend to that conversation. Um, or you are trying to attend to something without moving your eye, right? You're looking at a certain area, and you could imagine an athlete or someone playing poker or in that condition, like you don't want people to see where your eyes are, or you don't want it to be informative of your intentions. So you're looking at a certain area, but then you're covertly attending without moving your eye, without moving your fixation, you're covertly attending to some other area. Overt attention, on the other hand, would be cases where you're actually moving yourself in the world in order to get a better sensory input, in order to hear something better, in order to see something better. So it's really, um, it's tricky in a way that like we kind of mixed them up. Like in last lecture or last couple of lectures, we talked about attention in both overt and covert way. Like we talked about this sort of, when you were doing that let's say invisible gorilla experiment, you were moving around, you were overtly attending to a lot of this stuff. When we talked about in the context of bias competition theory, I said, well, you maintain fixation, so you're covertly attending to different objects for that experiment to work. Um, so we kind of mixed them up. I didn't really make that distinction. Both of them are under the umbrella of attention, but people, it's useful that you know the distinction between covert and overt attention. But it's not a complex topic in a way that, um, yes, it is possible to attend even visually without moving your eye. Like you could covertly attend. Um, so, as we talked about the anatomy of the eye and the visual system in general, if you're fixating at a point in a space, the area outside of that would be your peripheral area, right? So there is a central fixation area and you have the peripheral area. The fixation point, whatever visual input is at the fixation point, would reflect on the fovea, you know that. And the input that is outside, it would fall on the peripheral retina. So that's your periphery, like peripheral vision versus foveal vision. That's the terminology that we used before. So when you covertly attend to something, you're not looking at them with your fovea. You're not really processing it with your fovea. So it naturally means that you're getting some degraded information because it's not the best area in the retina to process it. We talked about this. Fovea is special for different reasons, and you studied this for the um, previous exam, where you have the highest visual acuity because you're close to the optical axis, because there is a foveal pit that opens up in order for the light to reach, and different reasons for also the this different distribution of cones and rods, but you know all that. But what is the result of that? Well, the result is that when you attend to something peripherally, you don't have the same level of resolution. So you cannot really detect objects that are really small. Now, this is one way to display it. If you all look at this A in the center, and without moving your eye, you probably can see what this letter is, right? Without moving your eye. But in order for that to happen, letter here had to be much bigger. This character had to be much bigger than what you're looking at foveally in order for you to be able to recognize it. So that's the point, right? That because you're averaging a lot of information in the periphery, when you attend to something peripherally, so that's what you did. When I say maintain your fixation on the center A and look at S peripherally, right? You're attending covertly to this S or attending covertly to this letter up here. And when you're attending to things covertly, because you don't have the high resolution of fovea anymore, it has to be much bigger for you to recognize. So visual acuity decreases with visual eccentricity. Basically, eccentricity is a distance from the fovea. How far something is from your fixation point is basically its eccentricity. Eccentric letters need to appear larger if they are to be identified as well as the fovea letter. That's the whole point I was trying to make here. I want to show you a demo of Do I have it? Okay. So look at this. A bee will appear, and I want you to track the bee. Okay, track the bee. All right. So you track the bee then, right? Now just keep your fixation on this uh, flower. So so you notice the difference, right? So when you track the bee, the point here is that when you're tracking the bee, um, as you go on, if I stop it here, you could see that these basically what the experimenter is doing, they're blurring the image with increasing eccentricity because they know that your fixation is at the bee. They could blur this flower in the periphery and you don't notice it, right? When you were tracking the bee. It shows that when you're tracking this bee, things in the environment could change in the periphery, but because you don't have the highest resolution, you cannot perceive it. Um, but when you keep your fixation on the flower, you could see, well, it's getting progressively more blur. So it's just a display of what happens to the information when they're in the periphery. And then when they become farther and farther from your fixation point, they become um, lower resolution. And that's what I mean. When I say lower resolution, it means that they become more blurry. Some of you experience that because you wear glasses and you know things look blurry without glasses, right? Um, but for people who don't have that experience, basically that's what it looks like. It's like you don't have the high resolution information. Like nobody has that information in their periphery. That's what I was trying to say. It's just a nice little demo, I guess. So the behavior, the eye movement behavior, is one that is the most common, right? We move our eyes 3, 4 times a second, right? So every time, like I'm looking, when you're reading, you're moving your eye. You're looking around, you're moving your eye, right? So that's a movement that we make a lot. And and if you remember, I said there are two low-level visual pathways. One of them is the genicular striate pathway. We talked about this. This is where the visual input goes from the eye to the LGM to the primary visual cortex. There is another pathway called tectopulvinar pathway, and this is the one we're going to just talk about a little bit today. In this pathway, the visual input goes from the eye to an area called superior colliculus, and this area is very important in the control of eye movement. And this area is really important because, let's say, our ancestors of lizards and frogs, that's their entire visual system, right? They don't have a cortex, which, which is the higher visual areas that we have. But what they have is basically visual input going to their, superior colliculus, and superior colliculus makes actions. Um, like for example, going for a prey um, or escaping from a predator or something. But that's their entire visual system. So some of the ganglion cells branch off and go to the superior colliculus, and then they also continue to the cortex. So in human brain or in monkey brain, the input from the superior colliculus ends up in the cortex and back, and there are a lot of recurrent activation and stuff like that. It is very important in making eye movements. And also, it has a retinotopic organization, and we're going to talk about that more. Um, it's an important area for integrating a lot of visual input in human brain or in macaque brain, where you want to decide where to look in an environment. It's not as easy as, well, the most salient thing, because we talked about it last time, that where you choose to look at to gather information or to perform a task depends on a lot of this stuff. It could be that it's very salient, or it could be that it's task relevant, that you're looking for something, that you're trying to gather information. But superior colliculus integrates a lot of those information in a map, which is retinotopic, and then it generates an action. So superior colliculus basically has, let's say, again, this is a schematic of it. But you could imagine people have found out this area is important for eye movement. How? Well, if they put an electrode in there and stimulate different parts of that surface, it causes eye movements to different areas of the image. So if, let's say, and a lot of this is done in monkeys. So if you have a monkey and then you lower your electrode to the superior colliculus area, and superior colliculus is really deep in the brain. It's not easy to reach it. It's not like some of the cortical area are somewhat easier to reach, but superior colliculus is deep in the brain stem. So you have to really go deep to reach it. But when you reach there, you could then stimulate it. So I don't think we talked about electrical stimulation that much so far. We always talked about only recording. But with an electrode, you could actually stimulate it. You could actually give it some, electrical current, and that would stimulate the cell and the surrounding area. It would basically activate them. They make them fire. And if you do that, you're gonna cause eye movements. So you zap basically a little electricity in there, and the monkey would make an eye movement with a certain amplitude in a certain direction. You zap it in another location, for example, and then the monkey makes an eye movement with a certain amplitude in a certain direction. So you could do that for the entire area in this. So this superior colliculus is really small. We're talking maybe 3 millimeters by 3 millimeters. It's not like, it's on that order. It's not like a huge area. Um, like a few millimeters by few millimeters. It depends on the animal, and it depends on individual, obviously. But it's a very small area. But you could move this electrode and stimulate different areas, and then you could basically map out what happens. Now, since this is a retinotopic area, if stimulating this part causes, let's say, upward eye movement of 10 degrees, and then stimulating this area would causes, horizontal, rightward movement of 10. What happens if I stimulate this area? Somewhere in between, maybe. What's that? Yeah, so because it's retinotopic, it means that it has an organization like a retina. So if two areas, if stimulating this area causes a movement in this direction, stimulating somewhere here causing this direction, somewhere in between should cause a movement in between. That's just, what retinotopy is, because if it was visual input, if if a certain brain area had a receptive field, another neuron had a different receptive field, a neuron between them would have a receptive field somewhere between them, right? Because it has to maintain the organization of the visual world. So this is a retinotopic organization in the action space. So if you take and stimulate this area, you would have a movement somewhere in between. So that's, people have done this. People have went out and basically did a lot of these stimulations just to map out what different areas do. So, but you see something peculiar here. So imagine this much tissue takes to go into, let's say, 10-degree. Well, why does it get smaller? Like, imagine that this is basically tissue of the brain. What happens here? That if I stimulate here, I get an eye movement that is like 30 degrees. So degrees here mean, so your surround is basically 360 degrees. So that's what it means. So you think of that way. So it's like 180, right? So basically, you could maybe move your eyes plus minus 50 degrees. So if I have a whole circle, half of it would be 180, half of that would be 90 degrees. And that's how much of the world you could actually move your eye in. That's what that means. So if I stimulate this part, I would get, let's say, an eye movement of 40 degrees. And then if I stimulate a very close by point, I get an eye movement of 50 degrees. So 10 degrees in difference. But here, in order to get 10 degrees of difference in the eye movement, I have to really move away in this tissue. What is that property? Magnification of what? Yeah. So what does this mean? It means that a lot of tissue is assigned to what? Yes. But in action space, what does that mean? What does that mean? Yeah, but in action space, if I say there is magnification of foveal area in the action space, it means what? What does that tell you about this tissue? No, you're on the right side. So it means that a lot more tissue is devoted to smaller movements of the eye. That's what that means. It means that you could actually make much more accurate movements of the eye when they're smaller, because you could make a 4-degree eye movement, a 5 degrees eye movement, right? And degrees being like, so if your arm is at the full length, your like the nail on your thumb is about a degree, let's say. So does that make sense? So this is 1 degree, and there are 360 of them if you go around, basically. Um, does that make sense to people when I say what does it mean to have an eye movement of certain degrees? Yeah, I'll say it again. So your surrounding is like 360. It's the whole circle. So half of that is 180. But the area in the world that you could actually have access to the visual world is smaller than that. So that's this much of this circle. So each of those, so basically there are, um, let's say in this case, the claim is that you could actually make eye movement. If you start from a center, you could make an eye movement up to this point, which is like 60 degree this way, or an eye movement this way, which is 60 degree that way. That's the claim. But cortical magnification, it means that a lot of the cortical tissue is assigned to making movements in this small area around the fixation. That's what it means. That you are prioritizing. Again, this is another trade-off in the brain, as always. You want to prioritize certain movement for certain other movement, and your central vision gets that priority, so you could make more accurate, smaller fixations than necessarily not than longer ones. Okay, so but eye movements are not coded by single cells, right? And this is an example that we see over and over again. They're distributed, and the way they distribute it is something like this. That's how people think about it. Um, let's say you have um, this is the surface of the cortex that I showed, and you could imagine that when you stimulate certain area, you're stimulating not just one cell, obviously, right? You're stimulating a big area of this tissue where you're stimulating that. So if you're stimulating this area, and I said based on the fact that these numbers are here, it tells you what that means. It means that you're making an eye movement in this direction. So when you're looking here, you basically move your eye here, right? But this is a collection of a lot of cells. A lot of cells are contributing to that movement. So the population coding is in that sense that you have many cells. When a lot of them are active, each of them contributes a little bit, they're voting a little bit, and then together they code for this longer movement. That's the population coding of eye movement in the superior colliculus.
What if there's damage to the superior cortex?
What is?
Yeah, I mean, would your eyes not be able
Oh, you could. So there is a lot. What? So the biological system has a lot of degeneracy, right? Meaning that there are a lot of different ways to do the same thing, right? In any biological system. So if you even remove superior colliculi, both of them from both sides, what happens is that you uh, still are able to make eye movements. There are like um, the frontal eye field. This is another area that controls eye movement, and it's in the frontal lobe. That's also connected to a lot of muscles that move eye movement. So that kind of fills in the place. It takes a little while, but people recover. Yeah. A lot of things that we say, right? I think that was a difficulty we talked about with lesion studies, is that you could lesion an area, but unless the lesion is really deep and big, you're not going to get that deficit. A lot of people, there are people who, are born without a cerebellum or some other area, but then they, move on and they have a normal life, um, just because I guess the brain can adapt itself. Yeah. Um, but but yeah, that's the idea. Now, if you're stimulating somewhere out here, you're stimulating a big area there, and then basically it's a summation of a lot of those activations of all of those cells that are coding for rightward movement to to get you there. Now, this should remind you, I'm going a little faster. This should remind you of what we talked about distributed coding in the case of recognition and perception, where having this distributed coding that you don't want one cell to code for one specific eye movement. That would not be a good system, right? This specific cell only codes for eye movement going here. This is a specific cell for eye movement there. But rather, you have a collection of cells. Each of them are coding for some rightward or upward or downward or leftward movement. And a population of them codes for one specific eye movement. And that, as we talked about it, again, this is a repeat of what we had before. Um, that is what we call distributed coding. And the benefits of that, you already know about it, but it gives you a more graceful degradation, for example, right? If some cells in the superior colliculus die, it's not going to make a big difference in your ability to make eye movement. And also, you could code them in a much more robust way. Um, so I want to show you a demo. And again, eye movement is important in many different domains, right? You read a paper in a sense that, well, it's a behavior you could use in order to see what people are attending to. And what people are attending to is a lot of time tells you, has information about their mental state or about a certain diagnosis or about a lot of other stuff. So in that sense, it's very useful for diagnostics and other purposes. And a lot of experiments you run and you study going to involve eye movement behavior. But I wanted to show you something. Another context that is really important is that a lot of the brain-computer interfaces stuff use these sorts of, they used to be more, did I get that? Brain control type. All right, let me see. I want to pull up one. All right. So this technology has been more developed um, because I'm going to talk about some other technologies that are not as developed in terms of brain-computer interface. But this one is much more developed in a sense that there are actual commercial tools out there, commercial, like the companies that are building these tools that you could use your eye movement in order to type. If you're locked in, if you can't move your eye, if you can't move your body or anything like that. Usually eyes are the last, like eye muscles are the last thing to go for people who have sclerosis. Um, so one of them is Toby. I couldn't find a better video, but this feels like an ad to some extent, but ignore that part of it. Um,
Just kind of taking this all in stride. So I don't think I really noticed the difference until we got the IVs. And at that point, it's like, whoa, okay, figure it out. His eyes looked tired, but he was smiling and just another man. And then
show how it works. So a system like that is basically you're typing with your eyes, right? Everything you're doing with your eyes. How does it work? Well, you have a keyboard and you're looking at a letter, and then there is a usually, let's see if it has an examples of it. So you see, you look at a letter or a character, and then there's a timer that goes off. If you maintain your fixation on it, it would mean that you want this character. You see, so you look around, you find what you want, you keep looking at it, it types, and then through that, like imagine some of these systems, if a person is really good at using them, could type, up to maybe 10 words, even a minute, if you are really good at it, and you practice it and stuff like that. And that's a lot, 10 words a minute. If you really sit at it, you could write a book in a year or so. It really changes people's lives when they can do this. So this person does, can even do impressive drawings with that. Yeah, so that's, I just wanted to show this example because a lot of I'm going to show later on today, I'm going to show some other brain computer interfaces that are really brain computer interface, that you want to interface the technology with the brain. Some of you read the paper on this that you had the assignment 2 for. But a lot of more developed technologies are based on eye tracking and stuff like that for people who are not able to move their body. And this, as I said, this is more developed. Like there are tools actually out there for it. Another area that I think studying eye movement is really important is in like um, expert attention behavior. Does anyone know what this is? Like what this image shows? No? Okay. Well, what is it? What is the image itself?
It's a part of the brain.
This is not, no. No, it's a lung, right? This is a scan of the lung, and it's like a cut of this direction. So, a radiologist, right? You take a three-dimensional scan of the lung, for example. This is not an X-ray, which is a frontal view. This is a three-dimensional scan of the lung, and then you are a radiologist would sit there, and then they would look through this volumetric thing, going forward and backward to see different levels, and then they would see what's wrong with your lung, if it's collapsing or it has if there's pneumonia or something like that, right? Now, in this example, there is something else that is very problematic. Some people, I'm sure, found it. Others also, right? Did you all find what's wrong with this figure?
Tumor.
Tumor? Could be, I don't know. There's another thing that I want you to find in that. If you find it, you'll find it, but it takes, yeah, right? Did you all get the gorilla? So there is a gorilla here. You see the gorilla? Does it show up right there? Yeah, it shows up. So this is from a paper called "The Invisible Gorilla Strikes Again." Now, what they did was that they basically took these scans that radiologists were going to study, and they just put gorillas different places with different levels of contrast, right? Intensity to make it easier or harder to find. And what they see is that, that many of them, almost all people, like, look at, and this is a true inattentional blindness, right? Because you don't expect something like this, and you're just looking through it, and you could look through all of them without realizing there were gorillas in them. Um, you could see that almost 80% of radiologists even miss, right, that there is a gorilla in there when they're going through this, because they're doing another attention demanding task, and they're not able to see the gorilla. um, it goes up to 100% in naive observers. So and it also depends on the level of intensity of how strong that contrast is. So I think this is another important area, an area to think about attention, and what, this perspective of cognitive neuroscience can help expert attention behavior. A lot of our jobs are basically expert attention behavior. uh, you're getting paid to find something that, or you're good at finding something that other people's aren't. um, so it's an area to study, and a lot of these concepts that we looked at kind of come up there. All right, back to this. So again, yes. Are there lie detection machineries? I don't know of any. Or you think that if someone's lying, their eye movement pattern would be different? Or they're looking up maybe like that or something like that? What's that? Natural linguistic processing? For lie detection?
For the way they look,
Oh,
if they look this way and I think they're lying or maybe not. I forget, but
okay.
it's like looking this way, this way, this way, this way.
Okay.
And you if it's kinesthetic, a memory that they're really, or if it's a lie.
Okay, then there is a study, or
I'm sure there is. I
okay.
look.
There must be something out there. Someone, huh?
What saccadic eye movement sprayed is human to see 2015, and they found that I can read the abstract, but basically they 30% participants expressed a true opinion and lied about another opinion within subject's design, and the number of saccades per second of speech was measured. As predicted, participants displayed fewer saccades when they told the truth than when they told a spontaneous lie. So.
Yeah, so that's an interesting topic for people to look into. If the Yeah, there must be other behavioral markers if people are lying or not. What's that?
They use pupillometry.
Pupillometry, yeah. So one thing they use is like you could measure the size of the pupil, and that a lot of time correlates with your emotional state. So people try to say, okay, if you're more nervous or something, then your pupil gets bigger. Um, so that's one measure that you could. I don't know none of these are reliable. There's no reliable. You could do a study and under certain circumstances find some result, but I don't think it's a reliable way of um, measuring lies, or finding out whether people are lying or telling the truth. Um, uh, yes, it reminded me of something, but I forgot what I wanted to say. Uh, the the attention network. So this figure is up here for me to say that attention happens everywhere in the brain. Again, I'm just talking a little about what we talked about before. It's not just one thing where it happens, let's say, in the ventral pathway and it makes things, receptive fields do something, or it's not one thing that only happens with eye movement in a certain area. It's a global thing. and in that sense, it relates to everything else that we care about in terms of your conscious experience or awareness or other stuff. So it's a network of areas that kind of coordinate, come to coordination, and then maybe dissipate. So and a lot of different brain areas are involved in it. um, I don't know if it's studying it at this level kind of is useful or not, but a lot of people do that in fMRI machines. They look at how these networks of areas emerge, where what areas are influencing each other's response and brain activity to map out, well, there is this frontal parietal attention network, or parietal and ventral attention network. So you just know that a lot of people talk about these concepts of attention networks, which is collection of areas that are talking to each other, something like that. And the areas that we talked about are kind of part of this, right? We talked about parietal areas being involved in attention. For example, if there's a damage to parietal area, you get these attentional neglect type of effect. Um, Frontal eye field that I mentioned, it's in the frontal area and it's involved in shifts of attention. You have the ventral pathway, obviously, doing all this visual processing. Attention impacts a lot of those things. And you have areas like superior colliculus where they are really involved in eye movement behavior. So we kind of looked at a bunch of these things. I don't know. I just wanted you to know that people talk about this concept of attention network as a network of areas. Okay, let's talk more about the motor control. Now, eye movement is a very simple behavior. It's an action involving a bunch of muscles around the eye, but it's not as complex of, let's say, hand movement or body movement or running or something like that. Um, and that also, as you could imagine, people have mapped out networks for that, where you have different brain areas that are involved in it. Um, if you remember, I showed you a video of what was the tool that a person moving around and stimulating. Okay, so we did talk about the stimulation, but it was a non-invasive stimulation. No, what was the tool? No, for stimulating. What's that? Yeah, so this is transcranial. This is TMS. You could move around, you could stimulate different areas, and then you could see the movement. And I showed you a demo of a person actually doing that. And you saw that, well, if this area gets stimulated, they move their hand. If a certain area next to it gets stimulated, maybe they move their shoulder. So that sort of a studies, again, lesion studies go with that too. You could use that study to see, well, this area must be involved in controlling this body part, and that area must be involved in controlling the other body part. So people have done that and found these different parts of the cortex. Um, this is the motor cortex being in control, supposedly, of different areas. Again, you see another types of magnification here, right? There is a lot more tissue assigned to your hands and to your face than, let's say, your legs. It's just that this dexterity that we have with our hands, it's very important to our survival, and like tool use, and more recently, I guess, writing, but that's not part of it. But back then it was just really this sort of being able to be so dexterous that we could use tools to do different things. Has really, right? So you would imagine brain would prioritize tissues for controlling your hands and face for facial expressions and those sorts of things. Um, how how good are these mappings? Not so good. Different tissues are involved in a lot of different things, but this just gives you a coarse idea of, well, these tissues are involved in controlling the hand, or these issues are involved in controlling the face. Um, a lot of time when people want to build this brain-computer interface, they put them in these dominant hand areas because they're bigger, and they're more dexterous in terms of the complexity of the neural behavior. So one way of thinking about how neurons can control action is, again, let's go back to this idea of selectivity. You could say, well, if a certain cell in my visual cortex is selective for a certain orientation of visual input, you could think, well, certain cell in your motor area could be selective for a direction of movement. In the superior colliculus, certain cell is selective for a certain direction of eye movement. Here, in the motor area, you could say, well, certain cell could be selective for a direction of movement, right? So if I move my hand from the center to this direction, from the center to this direction, right? From everywhere to this, in this direction, this cell could be active. So this cell, you could say, is coding for that direction of movement in my hand. So in this case, these are all recordings from a single cell, let's say. And each row is a different trial. Let's think of it that way. And what are each dot? What's that? Action potential. It's always action potential. When this is a raster plot, and each dot is an action potential. Each row is a trial, right? You're repeating this. What information do you get? And let's say this is all from a single cell. I put the electrode to a single cell, and I have hundreds of trials, the monkey moving it in certain direction. So what they tell you is that, well, here's, in this condition, this cell is really active. And this is a condition where the monkey is moving the lever downward toward in this direction. And the direction is shown as red. Here's a condition where the monkey is moving the lever in this direction and then the other direction. This cell is not very active when the lever is moving upward or up left or up right. Now, when the lever is being moved from this position toward the center, which is again downward, this cell is really active, right? Why do we have multiple trials? Well, you don't, one trial is not enough. You know, cell behavior is very noisy. You can't just look at one trial and make a decision about the behavior of that neuron. You have to have many trials, but you have many conditions here too, right? You're doing all these variations of where is the starting point and which direction it's going. From this starting point in the center, you have these, 8 different directions. From 8 different starting points, you could come to the center. So for all of those variations, you're just listening to a single cell and seeing what this single cell does. But for each of those, you're also doing it many times, so you can average them out, or you could see a better picture. When you see the behavior of cell plotted like this, what information does it give you? This cell is selective for, this cell is coding for what? What's that? Okay, so maybe this cell is most selective for moving in this direction, right? So this cell is active when the hand is moving in this direction, from somewhere in this direction. That's when the cell is active. It means that this cell commands or send that signal to the motor cells to execute this hand movement. That's what that means. Now you could imagine there is a cell for everything, right? This is, so the way we talked about the visual system, we said, okay, one cell is selected for this orientation of light. There's another cell that is selected for another orientation of light. So you could imagine that there are cells that are selective to all sorts of different orientation of movement now. So in this case, the neuron is most active, greatest density of dots, in this case, action potential, for movement in the downward direction. And those that have the highest activation, those movements that have the highest activation are shown in red here. And you could see that the cell is active for downward movement regardless of the starting position. Whether it's starting from the center or starting from one of these surrounding area, as long as it's moving toward the center, this cell is being active. So you could look at that and say, well, can I plot that more simply, right? You could say, well, instead of saying the cell is only selective for this, but the cell is also selective for this direction of movement, a little bit less, a little bit less. So you could show it as a graph like that, saying, well, this specific cell is most active for movement in the leftward direction, which is, again, imagine these degrees are starting from 0 here and going around you to become 360. So this would be 180. And the cell is most selective for movement in that direction. So when the movement is going in this direction, this cell is most active. But it also falls kind of gracefully. It's not like if you're moving in this direction, the cell is not active at all. You know, that cell kind of contributes to that. And that was also true of the visual system. We didn't talk about it, meaning that there's a graceful transition from one cell being responsive. If a cell, you kind of saw that, right? When a cell is responsive to a certain orientation, if you change the orientation a little bit, the cell is still a little active. If you change the orientation a little bit more, it's still a little active. So there's a graceful fall off of activation when you move away from the I guess the stimuli that the cell likes, or the stimuli that the cell is tuned for.
Do you have more cells when you're doing like fine motor skills? Like, does a neurosurgeon have like just greater selectivity in an area? I mean, like what kind of?
Sure, yes. I think expertise changes your motor, like it changes a lot of things. But people who do a lot more, dexterous hand movement would have a larger area, yes, in that controls their hand movement. So people have shown that this sort of plasticity changes the organization. So let me see. So let's take a break for 10 minutes, and then we come back, and then we talk about how population of cells move to control the movement. So now, if you imagine there are all these cells that are selective to a certain direction, right? Meaning that if you want to move in a certain direction, then how would you do that? How would the brain would make your arm move in a certain direction? Well, then that would have to come from a population of neurons coding that movement. But as we talked about, you don't want to have a single cell coding for one direction of movement. You want to have a population of cells coding for each direction of movement. So if, let's say, I was saying, well, if I'm moving in this direction, there's a cell that is coding for that is very responsive to that. If I'm moving in that direction, there are a lot of other cells that are not tuned for that direction to as extended as the other cell, but they are still a little tuned to that. So all of those cells could be active together depending on how well they match the direction of movement that you want to have. So let's say here, again, this is one way to display this. It's not the greatest way, but this is from the book. So if you're moving, let's say in this direction, right, upward, you could say that, well, a lot of cells would be active. But this would be what? A collection of cells. And let's say each of these brown line is one cell. and they recorded, let's say, from 200 cells. When you record from 200 cells, um, when you record from 200 cells as the monkey is making these movements, you could actually find out, you could specify what is the orientation selectivity of each of them, right? You could actually have a graph like this for every single cell, depending on how active they were for each direction of movement. Then you could say, well, if the monkey is moving in this direction, which one of those 200 cells would be active and to what extent, right? What do you expect? Well, you expect that the cells that are really tuned for that direction would be really active, and the cells that are not tuned for that direction would be less active. But a lot of cells would be active. Almost, most of those 200 cells would be active. It's just a matter of that they would be active to different degrees, and those degrees are specified by how much they match the direction of movement that you actually want to go. If you want to go in a certain direction, cells that code for that direction would be most active, and cells that don't code for that direction would be less active. Does that make sense to everyone? It's a population coding of movement. So the movement is coded by a population of cells, but the degree that each of them is contributing to that movement is different and depends on how selective they are for that direction of movement. So in this case, if you want to move in this direction and the dashed arrow is the eventual direction of movement that the monkey is going, it would be a summation of a lot of other smaller movement signals. What's a movement signal? Well, every neuron, if it's active, it's signaling a certain movement. This cell is saying, I want to move the hand in this direction. This cell is saying, I want to move the hand in this direction. Another one. But these two, if one cell, if you're here, and then you're being pulled in this direction and in this direction, you're going to go in the middle, right? So that's what this is trying to show you here. If you have different cells, different motor cells that are active, one of them is coding for movement in this direction. It means that when this cell is active, it's pushing the hand in this direction, or in this case, in this direction. Another cell is active, is pushing the hand in this direction. When both of them are active, it pushes the hand in this direction. That's what that means. It's basically two different vectors being added, but it really depends on relative activity of each of them. So a cell, a neuron, a motor neuron can pull your hand, pull your body in different directions, but it depends on how active it is. If it's really active, it's pushing you, pulling you strongly in a certain direction, then it impacts the average movement. You all see what this means, right? This basically, the size of these vectors is how active the cell is. The direction is how direction tuned a cell is, right? This cell is tuned for this direction of movement. It means that when this cell is active, it pushes the hand in this direction. That's the signal it sends to your hand muscles to move in this direction. So in order for the monkey or a human to move in this direction, it requires that 200 cells to be active in a specific pattern, right? That makes sense? It has to be active in a specific pattern where cells that are moving you in different directions cancel each other, and then what gets summed is eventually in that direction. So another way to say this is that each cell gets a vote, right? Each cell is voting to move you in a certain direction. These are motor cells. And motor cells send signals down to your motor neurons in order to move you in a certain direction. In this case, we're talking about the hand. So each of them gets to vote for movement in a certain direction. The strength of that vote will correspond to how closely the movement matches the cell's preferred activation, right? If you're moving in this direction, the cells that are most active are likely to be cells that are tuning for this, right? Because, well, you want to move in this direction. Those cells are selective for that direction, so they're going to be most active to pull you in that. And the cells that are selective for other directions, they kind of cancel each other out, and they're not as active. Does that make sense? How you have population coding of movement like that? How these cells collectively are moving the body or the hand in some directions. Does that make sense to everyone? So yeah, I guess we talked a lot about population coding in different contexts, and it's important, right? For you to know that, brain is integrated with a lot of systems, and we talk about brain areas, but those are part of networks. And in a brain area, you have this population of cells that are coding for perception or action. There's no difference, right? You want to have a population to do anything. So the motor network has a lot of areas, and I'm not going into any of it. What we were talking about is basically the motor cortex, which is up here, and there are a bunch of areas in it that are connected, and they're connected to your somatosensory cortex, where you feel and touch, and they're connected to the frontal area. So we kind of just talked about that. But there are, so the MC here shows you the motor cortex. That's where it's located. And you have other basal ganglia and other areas that are really involved in the movement, and obviously cerebellum, which is in the back of the brain and below the cortex, and that's really involved. And you have your, all the spines that are going down, and then all the way to go to your muscle. So it's a complicated system, and it's connected in a lot of different ways that allow you to move or to stop moving. But we're not going to get into any of those things. One thing we're going to get into is, now that we know, let's say, in the brain, in the motor cortex, now that we know we have these sorts of organization, a coarse organization. I said this organization is not super strong, but now that we know we have this coarse organization of different brain areas, and now that we know there are population of cells that could code for different movement, can we actually decode? Again, the concept of decoding, I'm going to get into a little more. Can we decode what the intention of the person is, right? What direction do they want to move? Even if they don't move, can we decode their intention? And can we decode what they want to do? And that would be something that you want to do in order to help someone who can't move anymore. So we talk about something like brain-computer interface. In this case, you basically have we talked about electrode arrays a few times here. This is basically what they look like. So this is a grid. It kind of looks like a bunch of nails that you put, but it's a really small thing. It's a really tiny thing, and it has so this says 1 millimeter. So the whole thing is 3 millimeter by 3 millimeter, and let's say in a lot of cases it has 64 channels, but there are bigger ones, 128 ones or slightly bigger ones. And what you do is that you can actually open up the skull and plant this. You put it right, you basically stick it into the brain, into the motor cortex, because the motor cortex is on the surface of the brain. It's right up here. It's on the surface of the cortex. So you could basically take that electrode array and plant it in the brain. And this is a MRI image showing you where it would be. And then you could see what those activities look like. You could record from different brain activity while the person is intending. Now, it all depends on the situation that you're studying. If you're dealing with a person, you could ask them to intend to move their hand, or you could ask them to intend to do something else, or to intend to speak something, right? if you want to use that to control a cursor or control a robotic arm, you put in this electrode probably in the dominant hand area. If you want to decode the speech, like their intended speech, you could put them in the areas that are controlling the muscles for the jaw, like the face areas. So these would be the components of a system like that. You have the person in this case, you have the electrodes that I talked about. And again, there are different ways to record brain activity. You know a bunch of them. It could be an MEG something, it could be an EEG, but in this case, let's go with an electrode array. So it's implanted in the brain and you're recording from the cells. The first step is signal acquisition. You're basically recording the signal. You're saying how active the cells are. Then you want to process that signal. You want to extract features from it, and you want to do pattern recognition. Now, this is very important. You want, if it's a person, you could tell them, just intend, and this is a person that say with, ALS or some other sclerosis, they cannot move their muscles, but they could intend to move their muscles. You could tell them, can you try to move your hand to the right? The hand isn't moving, but you record the brain activity when they try to move their hand to the right. Can you try to move your hand to the left? Can you try to squeeze your fist? Can you try to open it up? Can you try to move it forward? So imagine you have 30 different actions, and for each of them, they try to do that, let's say 100 times. At the end of that, what do you have? Well, let's say if you had, in this case, let's say there aren't that many cells. So these are raster plots again. And each row is a different channel in this, basically different cell. We're recording from a different cell. So imagine there, in this case, there are, I don't know, 10 of them. But you could look at the pattern of activation when they intended to move it upward, move the cursor upward, intended to move the cursor downward, right or left. And you could record that activity. If you do this many, many times, you would have a pattern now that corresponds to their intention of moving something to the right, that corresponds to their intention of moving something to the left. Now, in practice, you could then take that pattern and then connect it to a robotic arm or connect it to a cursor on the screen or connect it to one of these, elements in Pong, for example. You could play this. Then over time, the person can learn to, okay, so first of all, now if they want to move the cursor, they would do the same thing. They imagine moving up or they imagine moving down and the cursor moves. Maybe it doesn't, but the person in the loop can learn to think in a way that it moves the cursor up or down. But initially you do calibrate it a little bit to make it easier for them. But when you put someone in the loop, and it's an amazing, we're all very good at this. When you are in a loop that you could see the outcome of your actions, then you could actually change the way you think about it in order to change that thing. Right? You kind of learn how to, in this loop to make certain movements.
This is for people who have spinal cord injuries, right? Not brain injuries, but because they wouldn't be able to.
Yeah.
Right?
So this is all usually for people who have a spinal cord injury who can't move their body, or they have some other diseases that their muscles are, like in a sclerosis, they can't move their muscles, but their brain are still intact. Yeah. But the body-brain connection is severe. So I want to show you a few demos of these because I think saw that. One thing that's important is for us to be able to collect that data, right? We need to know what the neural activity looks like when the person intended to move upward, right? Otherwise, when later on they intend to move upward, we have to know, well, what does this pattern match to? Does that make sense to everyone? Why we have to collect that data? Because if we don't have the data and they're sitting in front of this system and they're intending to move upward, and we get a brain activity, we don't know that means moving upward unless we knew back then that this pattern means moving upward. But when you're working with a monkey, you can't ask them to intend to move upward or downward. So you have to do another trick. And we're gonna look at a monkey example first, and then we'll look at a human example. So this monkey had a surgery. They placed an electrode implant in the brain, and then this is a wireless system. So it's wirelessly communicating with the system.
The computer for a tasty banana smoothie delivered to your store. We can interact with the neurons simply by pairing them to an iPhone, just as you might pair your phone to a Bluetooth speaker. The links record from more than 2,000 electrodes implanted in the regions of Page's motor cortex that coordinate hand and arm movements. Neuron?
So they planted this electrode in a dominant hand area. In this case, it's the right hand for this monkey, right? And they're recording from thousands of neurons. And as the monkey moves the joystick, what are they recording? Well, they're recording the hand movement, but also recording the brain activity. So later on, they could match these two things. Say, okay, when the monkey moved the hand downward or upward or moved the cursor in certain direction, that was the brain activity.
Neurons in this region modulate their activity with intended hand movement. For example, some might become more active when he moves his hand up, and others when he moves it to the right. By recording from many neurons and feeding their activity into a decoder algorithm, we are able to predict Page's intended hand movements in real time. First, we calibrate the decoder by recording neural activity as Pager uses the joystick to move a cursor to targets presented on the screen.
So this process of calibration is the process of finding out what each neural pattern of activity among thousands of neurons mean. One pattern means, well, moving to the right. One pattern means moving to the left, and different meanings, right? That's what it means. That's what calibrating is.
We're wirelessly streaming in real time firing rates from thousands of neurons to a computer.
So this is the firing rates of all the neurons that they're recording at the same time, over time.
Using these data, we calibrate the decoder by mathematically modeling the relationship between patterns of neural activity and the different joystick movements they produce.
So now, after they've done that, they could unplug the joystick, but the monkey wouldn't know it because they're used to using it. They could unplug the joystick and then just basically see how the patterns, if they could just decode the movement from the electrical activity, from the neural activity.
We can use the output from the decoder to move the cursor instead of the joystick. Page still moves the joystick out of habit, but as you can see, it's unplugged. He's controlling the cursor entirely with decoded neural activity. Our goal is to enable a person with paralysis to use a computer or phone with their brain activity alone. Because they wouldn't be able to move the joystick, they would calibrate the decoder by imagining how. One of the things the Neuralinks allow Pager to do is to play his favorite video game, Pong. To control his paddle on the right side of the screen, Pager simply thinks about moving his hand up or down. We've removed the joystick altogether. Now that he's up to speed, let's increase the difficulty and see how well Pager can play with the Neuralink.
So in a lot of ways, so you see the process of building a brain-computer interface is that you want to have enough data to be able to learn that mapping between a brain activity and a certain control signal. And then when you have it, as I said, having this feedback is really important, right? living organisms are really good at exploiting feedback in order to make themselves better at whatever task they're doing. Um, so it's important that whatever you're imagining, you have to see the changes in the environment, and then you'll be like, okay, I know what I'm supposed to do more of. So you could imagine that a process like this could now be used to help people with um, with these sorts of, let me see.
Stanford University.
A spinal cord injury, people have a neurodegenerative disease, like this can be people who have a spinal. The goal of our research collaboration is to restore function to people with paralysis. This can be people who have a spinal cord injury, people who have a neurodegenerative disease, like Lou Gehrig's disease. We're able to eavesdrop in on electrical activity and slide a cursor across a keyboard and type out messages. Our research is focused on an area called the motor cortex. The motor cortex is responsible.
So again, similar process. In this case, you could have people actually intend to do something, right? This person is not able to move their body, but you could ask them to intend moving left, up, down, and then you could move this cursor, right? This is very elementary in that sense that you're trying to basically just give them the ability to communicate, right? It's a big deal. It's a big deal for them to be.
The body. Even in cases where you have paralysis, those cells, those neurons in the area of the brain are still active.
And you could calibrate them with a sample like this. So you could basically light up something in the periphery and they have to move the cursor there. Light up another point in periphery, they could, so it gives you information. Now you could record the brain activity while they're doing that. So that way you could say, okay, what is the pattern across these 1,000 neurons that I'm recording when they intend to move this way or when they actually moving this way? And these calibrations have to be done regularly because brain kind of changes a lot over days, and like we don't even talk about that, how much the brain changes. But that it does.
Those cells, those neurons in the area of the brain are still active. We can surgically implant a tiny electrical sensor that's made out of silicon that has 100 tiny electrodes that just sits on the surface of the brain, and it's able to pick up on the electrical activity of individual brain cells or neurons. We record tiny signals from those neurons, those brain cells, called action potentials. We can then take those signals and decode them using a computer. We can manipulate them once they're in digital form like any other data.
Okay, so I think the concept of decoding, you kind of understand what that means, right? When we talked about encoding, we talked about, oh, this cell encodes a certain orientation, or this cell encodes faces. It means that when you're perceiving a face, this cell is really active. Or when you're perceiving some other houses, this area of the brain is active. So it's encoding. Decoding is going in the opposite direction, right? You now want to decode what this activity means. Does this activity mean that they're imagining something, or does the activity mean that they intend to act in a certain way? So that's the concept of decoding. You could imagine something like that could be used to rehabilitate certain abilities, right? People who have had some spinal cord injury and they've lost connection between the brain and the body. You could imagine if you have a system where you're recording from the brain and then stimulating certain nerve cells in the body, you could potentially regenerate the connection between those cells, like the nervous system in the arm or in the leg or other parts of the body, and reconnect them because they're getting the same type of signal. People are working on that, being able to kind of redo that, or just basically move your arm, but short circuit it, right? If you directly go from the brain to a certain muscle in your arm, then you could move it and you wouldn't lose that muscles, right? You would use muscles when you don't use them. So people who get these spinal cord surgery, slowly lose all their muscles because they're not moving them. But if there is a way to short circuit that, go directly from the brain to that muscle, then you could continue using those muscles even a little bit in order to maintain them. So you can imagine that now, as I said, those eye tracking technologies are much more developed at this point. They're much easier to develop. They're much more consistent across subjects. In this case, you have to really record information from, again, when people talk about brain readings, first, I wanted you to know, have an idea of how primitive the state of the art is, right? It's not like we don't have anything near brain reading or anything like that. It's just you have to have an individual in the lab, you have to implant something, you have to record from them for hours and hours in order to get a simple mapping between their intention and some brain activity. And then you could decode some simple stuff. That's the level of things. It's not like some, like you could easily, and again, it's so individual, like you imagining something and someone else imagining the same thing, it would give you a very different brain activity. So you cannot take a decoder and apply it to someone else. All right. So one thing to know, an interesting thing to talk about are these concept of mirror neurons. And this is something people have found. In order to connect perception and action, they found that in this scenario, right, you have a monkey. When the monkey breaks a peanut or watches someone else breaking a peanut or sees someone, breaking a peanut but cannot hear the peanut, and then when the monkey hears but does not see someone, right? In all of these cases where they get a signal about a peanut being broken, this cell is active, right? So this is the idea of you have this holistic perception of an action, whether you're perceiving it, whether you're doing it yourself, whether you're hearing someone else do it. In all of these cases, you have certain cells would be active. And these cells are in the premotor cortex, and they're active both during perception and action. So it tells you, again, when they were found, I don't know, maybe a couple of decades ago, people thought they would be a big deal, that we have found these cells that are active during the perception of something and also acting on it yourself. But I don't think they delivered on that promise. It's just it just remained a small interesting thing for the most part. That there are cells that are active in all of these scenarios. But there's still people allude to them when they talk about the connection between perception and action, in a way that how can you maybe learn something from perception? How can you recreate something that you perceived, that you saw? Or I guess how your perception is dependent on your action means that your perception of things a lot of times is the way you would act on them, something like that. Like it invokes the same sort of activity. Another thing I wanted to say is we talked about this sort of, well, if you're in an environment, if there is an organism that is acting in the environment, depending on the visual input that you get, you're likely to want to have multiple choices. It's likely that there are multiple actions that you could do. It's likely that you are choosing between multiple things, and you want to know how does that selection happen, right? So a theory is that you have multiple plans usually in parallel. That your plans, when you perceive something in the environment, right, you have multiple choices and you want to go for one of them, right? There are two types of plates in front of you and you want to go for one of them. You're perceiving both of them, and both of them are going through your visual system. Um, and as they reach your motor areas, and then they reach your frontal area where the type of selection takes place, you slowly narrow down your options until you make a selection. Um, Basically, multiple action plans are made in parallel. Brain uses sensory information and sensory feedback to continuously update them, and these actions are competing with one another. Um, there is a nice experiment looking at that. So, so in this case, they're looking at the brain activity, and this is the experiment. They have two cues on the screen, and you're supposed to look at one or another one. But at this point, you do not know which one will be your target. So a fixation point comes up, two cues come up at different locations, and then they go down. So this is the working memory period of this, and we'll talk a little about memory next week. Um, you basically have two options, and neither of them are dead, right? You're maintaining both of them. So when you look at the cells that are coding movement in either of those directions, you would have both of them be active. As soon as you get a color cue that tells you, well, which one is the target, right? In this case, the red, the cells that are coding for movement in this direction, those are the selected. Then the other one becomes silent, and the coding for the red becomes much stronger. So this shows that when we have many options in the environment and we make certain choices, um, a lot of those options would be active. A lot of those options would be represented with different, with different cells. Different cells are coding for different stuff, and they're competing with one another. But then as soon as you make a decision, and this happens later on in the hierarchy of the brain areas, then that those cells become much more active, and through inhibition, lateral inhibition, they inhibit the other option, and then that becomes your choice. Okay, does that make sense? Right? This sort of, if you have many actions that you want to choose from, you could imagine this process. Um, and this process of, kind of, this is somewhat the perceptual cycle that I talked about. Now it feels like a long time ago. In a way that it's an organism in the environment. You have multiple options, you're perceiving them, you're sensing them, you're perceiving them. Maybe you recognize one of them, and then you want to go to that one, and then you act upon it. So it's like a cycle that an organism is in, and a lot of these choices are um, are uh, impacted by the kind of reward that you expect to get. Some rewards are immediate, like, if the options were between a salad and, mac and cheese, and you wanted to pick one of them, those choices are impacted by how much you expect to get reward from them immediately, or how much you expect to get reward from them in the long term. And that's what biases these options, right? In the example here, it was easy, right? The experiment tells you which action to take. But in real world, we have all sorts of ways to appraise our different options and then act upon one or another one. But a lot of things, a lot of context and emotional, responses and all those things go into that decision. Okay, that's it. I think we'll have